Hello everyone. So thanks again for coming to our user meeting. Like Mark mentioned, OmicSoft has been focused on for the last few years, and this is our current focus. This is going to be our future focus. But I guess for a lot of you guys, you know EncodeLand, you know EmcodeLand, and probably heard of GeneticsLand that I've been working on. But the key question is, what exactly are OmicSoft Lands? It sometimes seems like a database, sometimes it seems like a public data source, or sometimes you just feel, you provide a gene gives you some views. So I hope that after this short introduction and some updates and roadmap, we can have a unified idea of what OmicSoft is trying to offer land. So basically, we need to define what the land and the scope of the land, and then for the rest of the talk, we are going to focus on every single land. So we're going to talk about GeneticsLand, which will be officially launched today. That will be the second talk, and we are going to have very exciting updates on EncodeLand, some huge improvements on EmcodeLand and CVMLand, and then we'll also have these breakout sessions that hopefully will give you some live demonstration of the power of this type of land. OmicSoft as a company provides solutions for mostly enterprise users. We provide content, framework, and graphic interface. On the content side, mentioned EncodeLand, EmcodeLand, CVMLand, GeneticsLand, those are all essentially already been either has been released in use by users or will be released today. EncodeLand is actually a very interesting, a very special land that we're working on. I'll give you some details maybe later on. It will be part of this disease land. But it's interesting in that it actually has a very special data type that also fits very nicely into the NAND framework. On the framework side, we have NAND technology. Hopefully that will give you guys some ideas of what it actually covers. Project management, sample management, NGS, analytics. A lot of you guys are using our analytics platform, for example. Omics analysis, those are a lot of these advanced modules from previous microarray analysis development. On the graphic interface side, we provide project management, sample management. We provide pipelines for users, and we have more than 60 very interactive data visualizations that I guess everybody is using on a daily basis. Genome browser was very popular in OmicSoft software, and in the recent years, NAND visualization has been, I guess, the main user experience for, especially for those casual users who just want to go to NAND and find some sort of visualization. And I want to announce that we are working very hard on the web portal. The idea of the web interface would be, okay, we have so many lands that we can search. Can we have a simple place for average artists just to provide a gene or a gene search, or sometimes even a cell lab, and then be able to do cross-land search and provide a very simple user experience. So the first thing I would like to do is to provide an introduction to the NAND. NAND is a database for sure, because it consists of a lot of this public data and potentially your internal data. So it consists of 4 parts at this stage. EncodeLand is slightly different, and I will mention in a separate slide. But if we consider the first 3 lands that already been released, EncodeLand, EmcodeLand, CVMLand, we all consider this as genomics land. So I think you guys mentioned these are all gene-centric. Yes, you search for a gene or a set of genes, you get different data type of integration. So, but GeneticsLand is slightly different. On the world of genetics, everything was variant level. You can search for regions, you can search for genes, but then potential that gene essentially just was changed to a rigid brand. You can search for genes, SNP list, or maybe just a single SNP. So there is GenomicsLand and there is GeneticsLand. But the scope of the land was still trying to provide a centralized data management for these different data types. So land first is the data warehouse. For example, we are able to store unlimited omic data, unlimited VCF files. It's definitely a database in that VCF is a VCF file. If you want to do any modification, you have to create a new VCF file. So that's not a database VCF file. But anytime if you can do query, you can do updates, you can do add and remove, that's a database. Not necessarily relational database, but the ability to do this kind of incremental updates, I think in our word defines the database. Obviously, we have provided a lot of data, for example, TCGA, CCLE, this type of data service through NAND for our users. So it's a data service. We curate the data and control the vocabulary. We continue to do that. We have lots of updates today on that. So it's a data service for sure. But eventually, we hope to provide essentially two basic functions for the user. The first one is the ability to do public and internal data integration. Because a lot of you guys generate a huge amount of data, but the public data is always actually more abundant compared to your internal data. And some of you probably never generate any data, but still want to explore, what's available on the public data at a very deep level. Not say, just go to a website and trying to find some knowledge, but the ability to actually look into the data, basic point by point. That's the first goal. The second goal is actually to provide those advanced users, which I assume, allow you guys actually belong to the category that you guys belong to is to provide this kind of easy API access. Command line access to OShell. Our API has been quite widely used, but also starting with our web focus, the web API actually will be released earlier than the web portal, because the first thing is that we provide this web API, and then we develop those kind of web portals based on these API functions. And another, aspect to understand the land is to compare the land with project. We have local analysis functions, we have server analysis functions. Most of you are using Aristotle as a project basis. But then what's the relationship of project and land? So basically you have project 1, 2, 3, up to 1,000 or 10,000 unlimited projects. And then there are a few things that we need to take care before this type of project can go into NAND. Because NAND by definition is cross-project. So it has to do, anytime we need to do cross-project implementation. For things like somatic implementation, do we need to do it? Maybe not. But if it's things like arithmetic expression or analytic quantification, this type of normalization has to be taken care of first. But also, there's another angle of this cross-project metadata curation. So when we have multiple projects, they all need to have the same set of control vocabulary for the metadata side to be able to really integrate together. So NAND is cross-project, and you can think it as a project collection. Now we actually get to some really interesting kind of geometry or some intuitive ways to understand different ways of different products from omics on different NAND. So what really is the scope of NAND? So we try to work on structural omic data, not really just FASTQ files, PAM files, or not even VCF files. The goal of the NAND is to basically to fit everything into a two-way big matrix. So we have this way as variables, which I will define, and we have this way of observations. Most of the time, each observation is a sample. So it could be a copy number sample. It could be like a VCF sample from DNA sequencing. But sometimes this observation can also be a comparison. So it could be the difference of two groups. It could be an association study from thousands of GWAS samples. But then on the variable side, like we could have different types. Hopefully I will define next. But then each cell of this huge matrix actually represents some sort of information in the genomics or genetic side. And I want to mention that the number of these data points could easily go to trillions. So in the latest developments from OmicSoft, we define 4 different types of variables. You guys are mostly familiar with gene. So 30,000 genes or 60,000 genes depending on gene model. Those gene-centric variables most abundant, contains expression and copy number. It have actually all the somatic mutations for that particular gene. The second type is actually region-centric. For example, those peak values from EncodeLand actually is a perfect example of those region-centric variables. And if we fit that framework, that concept into this matrix, you will actually notice that maybe we'll get a sparse matrix out of this because different samples will have different regions, but that's okay. As long as it can actually fit into this two-way matrix, I think we can handle it as a NAND. The third and probably the most challenging part of variable type is what we call position-centric variables. Every single VCF file, for example, belongs to that category. You have a chromosome, you have a position, right, on the chromosome, and then you have some sort of values associated with this variable. It could be just a genotype along with some sort of quality. It could be a p-value, effect size, some sort of other information associated with some sort of association. But position variables, they are challenging because it could be hundreds of millions of them for a single sample. So the dimension is actually quite different from the gene-centric where we have a fixed number, usually between 30,000 to 60,000. The last one, although it actually sounds very challenging, so you guys probably are familiar with GVCF, right? GVCF actually contains the coverage information for every single position or 3 billion positions for every single sample. So there is a need to store this type of information because, I heard from people saying that we need to have the GVCF stored because when we have variants, we want to make sure it's actually covered, right? We see a false positive, but I want to be confident with this false positive by getting extra information. So even though this is kind of challenging, from the dimension point of view, it turns out to be actually much easier than the third type where the variants, the set of variants that each sample could represent is totally different from one sample to another sample. Because for the last type, you basically have a value for every single position. So just some examples of these variables. Protein expression could have the dimension from hundreds to thousands. Gene expression will be 20,000 to 60,000. Transcript expression will be probably double or triple depending on the gene model. Exon coverage will be another tenfold. Whole genome exon sequencing, 1 to 10 million. That's there. Whole genome sequencing, based on if you are doing joint or not, will be 10 to 100 minutes. Eventually, if we store the coverage, that will be 3 billion per sample. So now we actually define the scope of the NAND. It's a two-way big matrix, potentially very sparse. And we can actually have for each data type, we can have such a matrix. So we want to store this matrix, we want to import and export based on some common formats. We want to query the data in different ways, by row, by column, by subblock. We want to quickly traverse by row canvas by columns, sometimes maybe just a subset of this method. But since after we define this kind of geometry, things become easy to define actually the task that needs to be done. Visualization, very important, by row, by column, by block, analyze the data, integrate the data between different blocks. So those are the things that actually are behind this kind of oncology views that you guys have been doing. Those OncoLand or OncoLand or EmcodeLand or CVMLand. It's actually the same idea for the genetics. So by definition, land is cross-project and cross-data. But then another question is, we have two matrices, but really what does this each cell actually represent? It really depends. It could be a simple genotype. It could be a numeric value like gene level expression. It could be very complicated data types. For example, fusion. It could be, maybe a simple coverage, or if we're talking about those kind of encode data sets, it could have actually multiple value, like p-value, q-value. It could have those intensities. Whatever. I think the NAND itself does not define these data types. Based on what user needed, we can create actually data type on the fly. So the goals of the land, after defining this kind of essentially problem on the geometry side, we want to use this land to provide unlimited storage for omic data. And the second one is extremely important. I can say that the breakthrough of OmicSoft technology is that we actually need to compress the storage. I have a slide next. And that's a new update from OmicSoft. We want to provide fast incremental updating. So when you get a new sample, you don't want to actually create another database. This will not be the case for VCF, right? If you have a VCF file, you want to add a sample, you have to create a new VCF. Fast search and query, that's always be the case. Public data subscription, definitely important. So eventually we want to enable public and internal data integration. So easy access to for storing enterprise data. I want to spend a little bit time on this land technology because I said that we have a breakthrough, and this breakthrough is what we call the third version of land. So when we talk about this two-way matrix, I think there are solutions there, and people, when computer scientists think about this problem, the first idea is actually to store every single cell as a record and try to change this huge matrix into a relational database. I would say no relational database can easily handle, or maybe even theoretically handle, tens of trillions of data points, not to mention like efficiency. So that one was basically out. I don't feel that there is a real solution that I can store this kind of variant level data, or this large omic data at this type of level, at the relational database level. And the second idea is actually to do vector-based storage. There might be actually some sort of solution there. The idea is that instead of storing each cell as a record, we can store each variable that's most likely, or each observation that's less likely, as a vector, and potentially actually can even store as a blob with some sort of compression technology. This actually sounds good, except that I think there are two major problems. First, on the variant side, that space, like hundreds of millions of variants, that space is still too big, actually, for a standard database. The second thing is that it's almost impossible to do the updating. And we realized this problem 3 years ago, and we actually came up with a solution called Land version 2. I think that Land version 1 was always internal, so we never released it. You guys have been using, most of you guys have been using Land version 2 for the 3 years. So there's no problem. I think the speed is good. Sometimes you may feel, it's a little bit, taking a few hundreds of gigabytes of space for TCA, for example. Maybe when you try to republish, things are a little bit slow. But most of the time, there's no problem because we did a low-level optimization for incremental updates. And I think compared to Land 3, version 3, we have realized, we can still improve it so much. So this is the latest native development from OmicSoft, motivated by this GeneticsLand needs of storing random level data. As a result, we actually use this new version to store all the data types, including those region-based or gene-centric data. It's significant improvement on both sides and speed. This is a benchmark. Version 3 actually use only 1/6 of land to space. So basically, if TCGA previous take about 600 gigabytes, the new version will only take 100 gigabytes without losing any information. There's no double storage or storing both rows and columns. Publishing is 4 times faster. So even TCGA, maybe 3 hours, 2 hours, depending on your computer. And that's like that big. We fully support regions as part of this ENCODE effort. So later on, chipstick type of data can be stored into NAND. Fully supported GeneticsLand. So that's why we have a product that we will release next. Any query, even if just a simple RSID query, was actually optimized with thread pooling. It's guaranteed to be incremental updating. There's never a rebuilding need. It's a full database. And for GeneticsLand, where data could be huge, we actually allow you to use your cluster and lay down even your cloud to do parallel publishing. So we think that the publishing speed actually will be faster than, or even will be faster than your copying speed. So no matter how much data you have on the genetic side, we can actually incorporate into the land. So GeneticsLand will be launched next. I won't talk about too many details, but I want to mention that as part of this GeneticsLand, we're going to have 11,000 that's already been done, but then the goal is to finish all those 22,000 TCGA Gemini mutations as part of the land. And also the R API and Web API are also part of the land. So for the details and also a live demo, please see the next presentation. EncodeLand, just a few words on this. We have downloaded more than 90% of the ENCODE roadmap. This will be big land, but still size-wise is not very big. Sample size, it will be about close to 10,000 for human, and then we also have a mouse EncodeLand that actually guaranteed to be out early next year, but hopefully end of this year. This is a nice application of using the latest NAND technology to store region-based data, and this will be a part of the data land along with the very exciting single-cell land that will be described today. The NAND technology also accelerates a lot of this extra value that we added to existing NAND. So, for example, for EncodeLand, a few examples that how we use this new NAND technology to deliver this type of valuable data for the users. This clinical outcome land, which is a part of EncodeLand, actually collects 97 hundreds of samples with fully curated and fully controlled clinical variables, mostly on survival, but also other clinical variables. And we will have some updates on that, so that will actually give users some nice kind of data resource on the survival and clinical side. And EncodeLand, even though we have TCGA, sometimes people still want to have a second opinion based on arithmetic expression, so that's still very useful stuff. So so that's the introduction to NAND. I only have a few minutes. I will be a little bit faster on the updates, but everybody will get actually the recorded webinar plus all the slides. So OmicSoft just moved to a new building on April as part of our dedication to essentially analyze any public data as part of the land subscription. We are building our infrastructure to have 5 petabytes of data storage in 3 years. We already have finished 40% of them, and a lot of computing, a lot of internet and bandwidth. I also use Amazon as a backup computing, and to provide even better support for our users, we now have video tutorials, and we essentially improve wiki document to for every single function and every single option. We will have detailed information, so we're completely transparent to our users, thanks to the support team. On the framework side, we talk about version 3, and also the EncodeLand is essentially a totally different animal in this world, but fit nicely into our framework. We have some somewhat minor improvements, but still significant. For example, the RCM for Unlisted counting is going to be exactly the same as before, but 3 times faster. We have GWAT analytics, which will be demonstrated today live as part of the GeneticsLand offer. And also, we have another exciting talk this afternoon regarding on the DNA-6 side, how to incorporate the Syndicom GATK peptide, which will be 10 times faster, but also potentially seamless integration into our system. And also analytics for single-cell sequencing as part of the development of single-cell land. A lot of these details, I think, will be mentioned in the later slides, later talks. On the data content side, we also have a lot of updates. B38, you guys have been, asking for more than a year. Actually, we spent a huge amount of resource on B38. You don't have to have it, but if you're looking for both B37 and B38, we provide it. For example, even for a huge net like TCGA. I remember we have improved the land storage by 6 times. So even if you have B37 and B38, you still need like just 1/3 of the previous space. And we improved target and other lands. The cleaning outcome, X4O, those are all actually essentially available now, but as part of our regular quarterly updates, you will all actually get this update. On the GeneticsLand side, we have updates for both EncodeLand and GeneticsLand and single-cell land. That's being heavily developed at this stage. Eventually, if you are interested in and you have subscribed to GeneticsLand, you will also start to work with us on the EncodeLand, which will be used as well. So finally, this is my last slide. On the roadmap side, we're going to focus on 3 major things in addition to what we'll talk about those existing product, improving of the existing products. The first one is that we are going to this is for the GeneticsLand that we're going to improve our gene set management and analysis by closely working with you guys. The second focus would be OmicSoft Land. I know some of you actually probably don't have host your own every server. So, but even for those of you who actually host your data now with only like 1/6 of the previous space, it's still very likely that OmicSoft Land will be helpful for you because this will save space, and if you are interested in landing in 2014 and you don't want to store it internally, you can use OmicSoft Land as a backup. But also, I guess the most important thing is that if there is a data set that you guys are interested in, and we can build it mostly, upon request, and we can provide, instead of waiting for the quarterly update, we can actually provide quick access for this type of new land. And the last, but I guess the most important thing is the web API and web plans. That's really is going to change our user experience, and that's really the focus of OmicSoft, at least for 2016. Some data content, I think you guys are familiar, but we are going to keep improving B38 and provide parallel support for B37 and B38. Encode content will be a standard subscription, and more GEO and Express content allows more updates on the variant side. And other data types that we'll start to considering as it's not like we don't support them. It's like we do allow them on it for PCE, for example, but this will be like a standard subscription data types. So I think that's my talk. And up next, I think I'm going to introduce our head of genetics, and he's going to give us a formal introduction to GeneticsLand. Thank you.